everyone. On this beautiful March afternoon, bright and sunny, not quite typical for this time of the year for us, but I can see our speaker today has brought the sunshine along with her. It's my pleasure to introduce my friend and research colleague, Dr. Wendy McFadden-Smith, for our CCOVI lecture series. Wendy is one of our CCOVI professional affiliates, but many of you will know Wendy through her role with the Tender Fruit, with the Ontario Ministry of Agriculture, Food and Rural Affairs as the Tender Fruit and Grape Integrated Pest Management Specialist. She's also affiliated here at Brock University as an adjunct professor in our Department of Biological Sciences, where she also supervises fourth-year thesis students and graduate students as part of our program. For a little bit of background on Wendy, she has a Bachelor of Science in Agriculture in Plant Protection from the University of Guelph. Went on from there to get her Master's of Science in Plant Pathology, also from the University of Guelph, and then switched over to a PhD program in Plant Pathology from the University of Saskatchewan. Wendy's research focus is on the etiology of sour rot, grape fungal pathology, and of course integrated pest management. She's also a sessional lecturer in the Department of Biological Sciences here at Brock and also at Niagara College. And additional areas of research that Wendy focuses on is in integrated management of grapevine powdery mildew. I'm not going to say the official scientific name there. And the impact of powdery mildew on quality parameters of grape juice and wine. So please join me in welcoming Wendy for our lecture today.
Thanks for coming. I'd rather be outside in the sunshine too. I'm going to give you an update on what I've been doing with sour rot. As the sour rot queen, I figure it's my duty to my subjects to pass on this information as I come along it. Before I forget and before I run out of time, I'm putting the acknowledgements ahead. The Ontario Grape and Wine Research Incorporated funded the majority of this research. Niagara Peninsula Fruit and Vegetable Growers Association provided some seed money, and Vincor Canada and Schenck Greenhouses and Farms Limited were the sites for the last 2 years of research. I'd like to acknowledge my co-researcher, Debbie Ingalls, who keeps me on the straight and narrow and is my mind as far as the black box for biotechnology and anything that isn't in a vineyard, and winemaking as well. Christina Huber, who's in the audience, who's our PhD student. We've had a couple of undergraduate thesis students with this project. A number of summer students have helped with it, and other people in Debbie's lab. People from Ag Canada in BC have helped with developing some of the molecular technology that we've been using, and also Ag Canada in Ottawa. And Agricor has helped us with some of the sample collections. When they find sour rot in the field, will they flag it or bring it in to us so that we can do our thing with it. Also, a lot of the chemical companies have donated product for doing some of the screening trials that we've done. So this always makes me smile. Sorry. This is typical botrytis, what we're seeing now. A lot of the time, the berries will split, and you will see the sporulation of the botrytis on the surface. But also what we're seeing is another kind of rot, sour rot. And the first time that I really noticed this was about in 2005 when I was doing some powdery mildew trials in a Riesling block in near Vineland, and went to touch the clusters to turn them over to do my ratings, and they just kind of melted in my hand. The skins would split, and it was just juice. There was no pulp, there was no anything but a vinegar smell and a lot of fruit flies and yellow jackets. So I started watching this, and it was becoming more of a problem. So we decided in 2008 to start focusing some research on it. This is actually good botrytis. And once again, we have sour rot happening. So when you're looking in the field, botrytis tends to be more of a brownish color. Sour rot tends to be more of a pinky orange color. And the smell is the really diagnostic characteristic, that smell of vinegar that you can smell from the road if it's bad enough. On white varieties, it's more of a pinky brown color, red varieties, it's more of a pinky brown color. And we do see it in especially Pinot Noir. Why is it important? Because these organisms produce acetic acid. And this graph is showing just some data on sour rot severity in one of our sites, and then in green the relationship, the VA concentration. So you can see that there's a fairly strong relationship between the severity of sour rot and the amount of VA that's present. We don't know yet how much sour rot is too much sour rot. To a grower, any sour rot is too much sour rot. But as far as a level of detection, that's one of the things that we want to look at, is how much is too much? How much is tolerable? So there's a lot in the literature about sour rot, primarily in Italy and Germany, and nothing in North America other than sour rot that was caused by some fungal pathogens. This is documented more on the West Coast in California. So we figured we needed to find out what we were working with. Starting with Rhiannon's undergraduate thesis project, we started doing some isolations and developed a protocol where we would collect sour rotted clusters, extract, freeze them, then remove the berries by cutting the fruit stem, the the peduncle, and then surface sterilizing and isolating the organisms from inside those sour rotted berries. In 2010, this is some of the work that Christina did. She sampled 15 different vineyards, collected sour rotted clusters, and did these isolations. And what these bars show are the proportions of the total number of organisms that belong to each of these different species genera. So in this case, the green bars, say in this one, 89% of all of the organisms that were isolated from those sour rotted berries, they were acetic acid bacteria, either Acetobacter or Gluconobacter. The pink would represent Hanseniospora, the kind of yellowy is Bacillus, and the very pale blue is a yeast, Oreobasidium. So you can see that there is some variability among the different sites as far as the predominant species that was present. We didn't, hindsight is 2020, we didn't do sour rot severity evaluations at each of these vineyards to be able to relate the severity to the organisms that are present. That's one of the things that's on our to-do list. Just a couple notes. This first column here and this one here, the two that are marked with O's. Christina was really excited about these because she's a kind of an organic, biological kind of gal, and she was excited because these are Bacillus subtilis. But these are organic vineyards. Guess what's in Serenade? Bacillus subtilis. So these are organic vineyards that had applied this product. So this isn't a naturally occurring thing that's happening, this high proportion of the population. It's one of those things you have to kind of think about. The other thing that we have in this data set are two sites where we have the same grower with different varieties. So in the same area, different varieties, just to try to get some ideas as far as whether there's a difference among different sites and the different production methods. Christina did this again in 2011, and this time she started with 60 sites, 54 actually yielded, 54 or 55 actually yielded useful data. So here we have, once again, acetic acid bacteria seems to be the predominant species in most of the sites. Hanseniospora tends not to be such a big component, and these are Pinot Noir. And I'm showing three graphs. The first, the early season, Chardonnay is the middle, Riesling is later on in the season. So you can see once again that there is some variability as far as the proportion of the populations that are the different species. In this case, we also identified Candida, which is a yeast that has been associated with sour rot, and also Pichia in the yellow is another yeast that has been shown to be associated with sour rot. As far as the pathogenicity, we're still trying to get a read on that. So more of the same. This one is kind of interesting that it's such a low proportion of acetic acid bacteria and a lot more the Oreobasidium. So there's a lot of data mining to go on with this yet to try and determine relationships of location, relationships of the spray regime that was applied. Perhaps there are products that were applied during the growing season that had some effect that we don't know about. So we'll be mining a lot of this data. And Riesling. So once again, the same kind of story. These were just isolations. Now you'll notice that Some of these are not 100%, so there's other stuff that grows there too. Different yeast, different bacteria that had not been reported in the literature for being pathogenic. So Christina developed a method of evaluating sour rot severity. She inoculated berries and developed a scale from 0 to 4, which describes the extent of colonization within a certain period of time. So 4 would be the most pathogenic, 0 would be not causing any disease at all. So comparing as a mean over these different organisms that were isolated, both Gluconobacter and Acetobacter are giving pretty severe sour rot symptoms. This is with a wounding, so the organism is applied to the surface and then it's the berry's wounded and then the organism is applied to the wound, right? I don't know, she did it.
Depending on whether it's a suspension or whether we were just rubbing the color.
It was with wounding anyway. It wasn't direct penetration. There was some kind of injury to the fruit. So Acetobacter, Gluconobacter had quite a bit of pathogenicity. Hanseníaspora, still pretty good. Candida xamplanina, not great on average. And the complex was a mixture of all the organisms together in a soup, and it's not a lot better than the Acetobacter. So there's no additive or synergistic activity here. This graph shows the variability in the pathogenicity of the isolates that Christina isolated. So with the acetic acid bacteria, they're pretty much all uniformly pathogenic. Hanseníaspora, there is a little bit of variability. In Candida, there's a lot more of a range. So this is another aspect that Christina will be looking at. Why does it happen? We've got a pretty good idea now that the acetic acid bacteria and Hanseníaspora are the two main bad guys. Sour rot's found in varieties that have tight clusters and thin skins. Normally we'll see it start in Baco. Baco's the first one to go, then the Pinots, Gamay, Chardonnay, Riesling, Gewürztraminer. We have seen it in some of the red varieties, Merlot, Cab Franc, Cab Sauv, depending on what happens in the season. But generally we'll see it most severely in the earlier varieties that are thin-skinned and tight-clustered. The sour rot organisms, when Christina did her inoculation, she wounded the berries. They haven't been documented as being able to cause disease without any kind of way of getting into the berry. So any kind of wound that you may have will allow them to get in. So some work that was done in New York showed that it's possible to have superficial infections of berries that you can't see with the naked eye. This is an image of powdery mildew fungus trying to penetrate grape berries, and all of these little brown spots are places where a hypersensitive response has happened, where the fungus has tried to invade, the cell has died. But these are also wounds. So what Gadori and group found was that these are two apparently healthy clusters, but when they're incubated, this was the kind of thing that they found. And they did some isolations, and they found Oreobasidium, Hanseníaspora, Candida stellata, which is now considered Xamplanina, and they did find acetic acid bacteria, but they didn't identify which ones. For the longest time, sour rot was considered to be part of bunch rot, part of Botrytis bunch rot. But we've now determined that you can have sour rot without bunch rot, you can have bunch rot without sour rot. This is very typical, beautiful powder, Botrytis sporulating along the splits in the berries, coming through the lenticels in the berries. I just like that picture. I ran correlations over, we've got this huge data set with multiple treatments with ratings for both sour rot and botrytis, ran correlations on them, very, very weak correlations. The highest correlation coefficient I got was 0.147, so basically there doesn't seem to be any relationship between these. Grape berry moth. Botrytis, sour rot frequently found associated with grape berry moth injury. It's another wound. However, we find sour rot in the absence of grape berry moth injury, in the absence of bunch rot, of Botrytis bunch rot. Fruit flies. I was accused of planting this fruit fly here, of finding a dead one and gluing it on the berry. This is a candid shot. I really took this. It was alive. It flew away shortly afterwards. Fruit flies. One of the diagnostic characteristics of a sour rot infestation or infection. You can just imagine this maggot squirming in these berries. Sour rot berries are completely full of these maggots because the fruit flies like them. And often when you go into a vineyard, this is the type of injury that you'll see, a hollowed out berry. No frost the way you'd see with great berry moth, just the whole guts of the berry are gone. So sort of a chicken and egg thing. What comes first, the sour rot or the vinegar flies? Do the vinegar flies bring the sour rot organism? Does the sour rot organism attract the vinegar flies? Yes, is the answer. We don't know is the real answer. Vinegar flies can vector the sour rot organisms. They can carry it on their body parts. When the eggs hatch, they're usually laid along the juncture where the berry skin attaches onto the receptacle, and as the berry starts to increase in size, it will pull away. The fruit fly knows this, lays its eggs close to that juncture so that as soon as that berry starts to expand, there's a way for the larva to hatch out of the egg and go into the berry. So they're very intimately associated, kind of the same as botrytis and great berry moth. Yellow jackets. Yellow jackets will eat berries and cause injury. Another possible chicken and egg scenario because they tend to be wherever there's sour rot. We don't know whether they're carrying it. One would assume that they would be, or whether they're just attracted by the sour rotted berries. One of the things that I noticed is, here's an example of the way that the sour rot develops. It generally tends to go from, if it's a visually uninjured berry, from where the berry attaches onto the peduncle into the berry. Weather. 2009, Christina's first year. It's testament to, if you want to control something, start graduate work in it because it won't be a problem that year. It was very, very cold during harvest. Very little sour rot developed. And we had frost fairly early. She did monitor temperature within the vineyard. Blue bars are the rainfall. Purple is the Brix accumulation, and red is sour rot. This is just incidence, it's not severity. But what you can see is that it wasn't until October that we really had an increase in sour rot, that it really started to swing up. And that was after some rain and when the temperature started to increase. So there's a temperature relationship there. We don't know what it is yet. 2010 was a great year. It was a great year for grapes. It was a great year for sour rot. Once again, monitored temperature, precipitation, and Brix accumulation. And what I want you to look at is around what Brix sour rot started to increase, started to upswing. Between 13 and 50. You'll see that in the next slide as well. Here we are in 2011. Decided to measure TA as well. This is in the Riesling block. Once again, that upswing, look at the Brix, between 13 and 50. And the Pinot Noir, between the 13 and the 50. So there's a temperature thing going on, but there's also something changing in that berry that's changing its susceptibility to infection. So Christina decided to look at this. This is one of our incubation chambers. For this test, she was looking at Pinot Noir. She went out. Pinot Noir develops a lot of secondary clusters, so you can get a range of different maturities of berry at the same time in the vineyard. So she went out and collected berries that were at about 9.7 Brix. She just did by color, that she would take some that were green and then squash them to determine the Brix, and then use that as an example of what the Brix would be. Some that were at 13.4, some that were at 15.5, and then she did 11.2 as well, and looked at disease development. So here's what those berries looked like after they'd been incubated for a little while, and this is the disease progress. So you can see a big jump between 13 degrees Brix and 15 degrees Brix. We'll be looking at this a little bit more as well in the future. What can we do about it? Two strategies. The first one is try to reduce the injury to the berries so that there isn't a way for the sour rot organisms to get in. The other one is to get rid of the organisms. As far as reducing berry injury, one of the things that we see frequently is that sour rot will develop in the center of a cluster. So as those berries are in stage 3 and they're starting to rapidly increase in size, those berries on the inside will get squashed. And the sour rot organisms that were on the surface of the berries can infect. You'll also see it in other areas where there's berry injury. So a couple strategies. The first one, reduce the amount of berry squeeze by loosening the cluster. Secondly, make the cuticle thicker. Make the skin stronger so that it's not going to be as prone to splitting. So we used a couple, there's a US conversion that I didn't change. Fruiting zone sprays at about 600 liters per hectare. We applied gibberellic acid because it's known to be able to change the architecture of the clusters. It's used in table grape production to stretch out the cluster and allow the berries to get larger. That wasn't our goal, but to stretch out the cluster. We looked at 5, 10, and 20 ppm, applied at pre-bloom, 50 to 80% bloom, and then 7 days after that. We looked at another product that's been registered in Europe, prohexadione calcium, at high, medium, and low rates that I'd have to kill you if I told you what they were in metric. And Stimplex. Stimplex is an extract from seaweed that is supposed to have some activity against bunch rots and have some effect on loosening the clusters. So we applied that at 80% bloom in one of the years. Zabadol and Dittmer in Michigan developed a cluster tightness or cluster compactness scale, 1 being the tightest cluster possible, where you can't even budge the cluster, you can't move it at all, to 6 being where very few of the berries are in contact with each other. So each of these clusters was evaluated for the cluster compactness. The other strategy that we used was one that had been described in Italy by Dr. Pony, also in Michigan by Tom Zabadol, and in Pennsylvania. Brian Head had taken a look at it. And this was the strategy of removing the leaves before bloom, the basal leaves before bloom. So starving that cluster so that fewer berries would set, so that you'd have fewer berries to squash up against each other. This is what the before bloom leaf removal would look like. This is in Pinot Noir. How's that for ampelography? This is after the bloom leaf removal. So you can see we took the basal 6 leaves. At Veraison, this was untreated with no leaf removal. This is Pinot Noir again. This is leaf removal at bloom. So you can see that there's still a lot of leaf regrowth in that fruiting zone area as a result of laterals breaking. So there's enough leaf or cluster exposure to be able the clusters to dry, but enough leaf coverage so that they don't suffer from sunburn. This is pea-sized leaf removal, so you can see it's a little more bare, and this is Veraison leaf removal. In 2009, we saw a lot of this, especially in the reds where we were struggling to get them to mature. So looking at the effect of bloom treatments on cluster looseness, the only one that significantly increased cluster looseness on the Riesling was prohexadione calcium at the high rate, and also in the Pinot Noir. I think our timing was a little bit off with the 2009 applications. Sour rot wasn't a problem, so we just looked at incidence, and a lot of the treatments had a significant effect on sour rot incidence compared to the untreated check. In 2010, no difference among the treatments as far as cluster looseness or disease severity. 2010, we went from pre-bloom to bloom over the long weekend. It wasn't the long weekend actually, it was earlier than that. So it was a very quick transition. We couldn't get that 50 to 80% bloom. It was just the timing of these treatments is so critical. If you miss it, you don't get it. And similarly for Riesling, we missed the boat. 2011, we got it right. Here we're looking at sour rot in the light purple bars, botrytis in the dark purple bars, and cluster looseness in the yellow. This is in the Riesling. And several of the treatments resulted in reduced sour rot. GA at 5, applied once or twice, GA at 10, GA at 20, and the prohexadione calcium at the high rate. So this was promising, this was encouraging. You always want to have something that works once in a while. There is no journal of negative results. The interesting thing, though, is that those lower, not all of those lower disease values were associated with increased cluster looseness. So there's something else going on. I don't know what it is. We couldn't pull out these differences statistically at the Pinot Noir site at harvest. However, the grower called me in October and said, you need to come out to the vineyard. He's a really great grower. He's a really great cooperator. He didn't pick these clusters. He left our plots unpicked because he wanted to see what would happen. And what he noted was that 3 weeks after commercial harvest, the plots that had prohexadione calcium at the medium and high rate and the GA at the 20 ppm had significantly less sour rot, and botrytis didn't seem to make a lot of difference, but the sour rot was really knocked down. So that's kind of interesting that it had that effect. One of the concerns with using these hormonal products is the effect on return bloom. We looked at return bloom between 2009, 2010 and 2010, 2011 and didn't find a lot of difference. Now, in talking about this with Dr. Pony, I made a mistake in my randomization and I re-randomized. And what I should have done was applied the GA treatment on the same plots year after year after year to see if there's a cumulative effect. So I'll try it again this year and see if there's a cumulative effect at least between the 2 years and see what happens. Reducing berry splitting. A lot of the information the ideas that I had for reducing berry splitting came from the West Coast for materials that are used for reducing cherry splitting. Berries and cherries, cherry splitting is a really big problem. In Ontario, the 2 times when you can always count on rain are cherry harvest and grape harvest. So I took a page from the BC and California and Washington group. Did not hire a helicopter because I didn't have the budget for this. It's really amazing to see this. But I have some ideas of other things that we might be able to use that I'll be looking at this year. And you have to come and work for me if you want to know what they are. Some work that's been done with the cherries, looking at salt solutions to reduce the amount of water that moves into the cherries by adjusting the water potential outside so that it's equal to the water potential inside, so the water doesn't move through the skins. Applying calcium to strengthen the cell walls, and applying surfactants so that the water will run off the surface of the berries. They don't stay wet as long. The conditions for proliferation of the pathogens aren't as extensive, and also splitting is supposedly reduced. So we looked at 2 different types of surfactants. So the calcium product that we used was Stop It. It's registered. It's the only one that I could find that had grapes on the label. We used a 12% weight by volume solution. We did leaf removal at either pea size, berry, or at veraison, and then we applied rainguard or Desicoat. And the calcium applications were made either twice at pea size berry and 2 weeks later, or at pea size berry and weekly for 4 weeks in a row. The total amount of calcium that was applied was the same. What we found was the only thing that really reduced, that had any effect, was the pea size leaf removal relative to no leaf removal. The rainguard seemed to do a pretty good job on the botrytis, so that's kind of something to keep in our back pocket as another idea. That was Pinot Noir. And the Riesling, nothing seemed to really make a difference. This vineyard was really whacked. It's a great vineyard to work in, but things got out of control. Now, we also did some work with this large grower on this he's actually a small grower, with this cooperator in some large plots where he did whole row treatments for us. So we looked at botrytis severity with these different, either no calcium, 2 calcium, or 4 calcium, pea-sized leaf removal or veraison. And then we looked at the mean severity of sour rot, and we also looked at the percentage of clusters that had sour rot more than 10% affected, and the incidence of clusters that had more than 25% of botrytis. Those were kind of arbitrary cutoff guesstimates as far as what would be commercially acceptable. And what we found was that for some reason, the 2 times calcium seemed to promote disease. I have no idea why, it makes no sense at all. But this is the big story, the pea-sized versus veraison leaf removal. Major difference in almost everything except for the percentage of clusters with botrytis over 25%. So the moral of that story, get your canopy cleaned up early. Don't wait till veraison to do it. And it'll be worth it. As far as adjusting the population that's on the surface of the clusters, potassium metabisulfite was something that I'd read about and in Australia and New Zealand, that they tried it as a vineyard application to control botrytis. It's not conventionally used that way. It is a food-grade product that's used in the winery to reduce oxidation and eliminate unfavorable yeasts for the vinification. I took from this literature the rate, 5 kilograms of potassium metabisulfite per 1,000 liters of water, because there was no phytotoxicity reported in the literature. So I figured that's a good place to start. Didn't work really well in botrytis, by the way. We didn't know whether it would work. We didn't know how much to use. We didn't know when to use it. We don't know how it works. And we didn't know whether it would have any impact on vinification in the end, whether there'd be any residual SO2 issues. We're not going to cover the residual SO2 issues in this one because I talked about it the last time. Let it be said just that there were no residual SO2 problems. There's no impact on vinification. There was no H2S issues, even with 1 day of pre-harvest interval. So our post-veraison treatments, potassium metabisulfite at low, medium, and high rate. We looked at millstop, potassium bicarbonate, that had been reported in BC to have some impact on the yeasts. We did a combination. We looked at oxidate, which is a peroxide product. Blight band strain A506 is registered in the States for sour rot. This is the kitchen sink approach, the shotgun approach. Actinovate, or blight band contains a biological control organism, Pseudomonas fluorescens. Actinovate, Streptomyces liticus, another biocontrol organism. We were contacted by a company who was looking at registering oxysilver nitrate as a sterilizing product. So we took a look at this. And we even looked at vermicompost, so the soup that's the result of worms eating stuff in a worm farm. Applied at 6 liters per hectare in the fruiting zone, started at about 50 to 75% veraison. 2 weeks later, put another spray on, a week later, and so on, trying to guess when harvest is going to be. We sort of have a little bit of experience of that angst of a grower of knowing what the pre-harvest interval is, because you can't extrapolate when harvest is going to be. So we sort of had to arbitrarily decide a little bit. A couple more sprays with the Riesling because the season was a little bit longer. So in the Pinot Noir, once again, sad news. None of the post-veraison treatments seem to have much of an effect. The Agress looks pretty good, but there was a lot of variability, so it's difficult to pull out the differences. That was the oxysilver nitrate. The KMS knocked it down some, but this is your check, the pea-sized leaf removal, because pea-sized leaf removal was done throughout these plots. So in that case, all of these are not significantly different from our check. You can see the benefit of doing leaf removal at pea size versus none though. The Riesling, our star spot again. Here you have our check, the pea-sized leaf removal. So looking at everything that has red arrows, these were all significantly lower in sour rot than our check. So blight ban, the biocontrol organism, aggress at 2 of the 3 rates, KMS at applied 6 times, at 5 kilos per hectare, and at 10 kilos per hectare. These all reduce the percentage of sour rot. The other thing that we were trying to do was identify when do we have to spray. Do we have to spray 6 times? Crap. So we looked at 1, 2, 3, 4, 5, 6, 7 different treatment timings, from as much as the time from the beginning of veraison, so you're looking at about 2 months, 2 weeks after veraison, 3 weeks, a week pre-harvest, 3 days pre-harvest. This one was sprayed both 1 and 3 days, and this one was sprayed 1 day pre-harvest. So there isn't a nice progression of increasing disease, so we haven't nailed it completely, but at least we have some results that show that it's knocked it down, so we can start to play around with this a little bit. And the effects seem to be the same against botrytis as they were against sour rot, with reducing it, or a little bit better. This is just to show you what a trooper Christina is. These aren't all her plots, but she did help me harvest all of them. This was quite a team effort. We had 40 treatments replicated 4 times from each plot. We collected 25 clusters and rated each one. So we are the sour rot queen and princess. What are we looking at in the future? I was told by my grad supervisors that if you're a good researcher, your research always generates more questions. You never find all the answers. All you do is find more questions. So some of the things that we want to look at, the effects of temperature and wetness duration on infection. We're going to try doing this in incubator studies with inoculated fruit. Looking at some practical applications. What's the importance of soil moisture? Some research in New York has shown that vines that have high moisture pre-harvest tend to be more prone to botrytis infection. So maybe there's something going on there. Can we manipulate this? Is there effective soil type? Are heavier soil types more prone to sour rot? I've heard pros and cons. I've seen our Pinot Noir site is on very light soil. Our Riesling site is on very heavy soil. They both get whacked with sour rot. Vine vigor, all different things. The use of cover crops, manipulating the vine to be able to reduce sour rot. This changing of susceptibility in the berries between 13 and 15 Brix. This is one that really intrigues me, and I'm really interested to see whether we can get the same kind of thing with botrytis and try and tease that out. Christina's going to be doing her PhD for the next 8 years. There are other potential biocontrol agents that I'm looking at. This isn't part of Christina's thesis. I'm going to try this repeating the hormonal treatments and the leaf removal on the same plots that I did them on last year, and see if that has an effect on return fruitfulness and whether there's an increased effect if it's cumulative, doing it year after year. One of the questions that growers ask me all the time is, does KMS work when it's cool? Are we relying strictly on the volatilization of it, or is it the SO2 in solution? Does that have an antimicrobial effect? And my suspicion is the latter, that SO2 in solution is also an antimicrobial. The interaction between causal organisms. Some of the literature has described sour rot as a stepwise process, that it's not just one organism that's involved. So Christina will be screening these organisms, inoculating one, then inoculating the other, or in reverse order, inoculating them together, and just seeing what she can get. And finally, we're going to look at the progression of berry microflora through the growing season, from pea-sized berry through to harvest, just to see if there's a shift in that microflora through the season, that the sour rot organisms tend to multiply up before we actually start to see sour rot. Maybe it'll give us some ideas for manipulating it and controlling it. And that's it. Thank you.
Wendy, I think one of the questions that I had that you you started to, on one of the slides, I think articulate some of the issues that we have with sour rot research is just the variability we get from season to season, especially with the rainfall, and then also starting to look at some of these linkages with the Brix development. But for some of the sprays that you're using, rainfall may also have an effect of washing some of the sprays off. Could you comment a little bit on that?
Well, KMS especially is not formulated to be used as a pesticide, so it has no sticker, it has no spreader, so that could be something else we could look at, is trying to increase the retention of KMS. I don't know whether it would retain its activity, how quickly it will dry, whether once it dries it's still going to be active or not. The microbiologist who know that. As far as some of the other ones, they are formulated already, so they should have stickers and spreaders in them that will extend the activity of them.
Well, on that note, if we stick, now sulfur too is a potential to have berry damage from the sulfur, kind of like when we use oil and sulfur in combination.
So
I'm going to let Debbie handle that because I always have trouble with the sulfur thing.
you're talking about different forms of sulfur, so what you're talking about is elemental sulfur. What the KMS is, it's sulfur dioxide, so it's a different form of sulfur that is in a completely different oxidation state. So still, it's something that we would want to check if this ever ended up being a treatment that looked promising for the vineyard. But again, just don't confuse elemental sulfur, which of course nobody would want to spray one day before harvest because your winemakers would kill you because you would have lots of H2S, rotten egg smell in your fermentations, versus sulfur dioxide, which is an oxidized.
We're co-researchers, this is legit. Max.
Was KMS acidified and other trials?
We tried it acidified and straight and didn't make any difference. I think the straight actually worked better than the acidified. Somebody else had asked about using potassium sorbate at one of the places that I did this presentation, and I don't know whether it's the SO2 part that is the active part, not the...
Potassium sorbate is an antifungal agent used in winemaking. There's residual sugars in wine, so I guess that question would come from some of the wild yeasts that are associated with the sour rot complex, but not something that we tested.
Try it.
Yep. Question from an online viewer is, in our vineyard, we induce botrytis for a delicious botrytis-affected wine. Have you seen any products or techniques that can discourage sour rot without affecting our noble rot? I.e., are the temperature, humidity, grape wetness conditions different for the different rots?
Doesn't seem to be. It seems that when we have weather, first of all, the online inquiry seems to be from some place that doesn't have rain at harvest, because we don't have any problem getting botrytis most years. We have a problem with too much botrytis. But as far as the conditions, generally, if it's a year for botrytis, it's a year for sour rot, even though they don't have a good correlation. Thank you.
He says they're in BC.
I'll be talking to you this summer.
There's another question as well. Sorry. As a grower, what are the early conclusions that can be used in our strategies? That's from Fabian Doggy.
Do your leaf. So the question is, what can a grower do?
What are the early conclusions?
The leaf removal at pea-sized berry as opposed to veraison is probably your strongest strategy, making sure that you have a good open canopy. When you go to do your cluster thinning, make sure that you don't have clusters on top of each other, because that's usually one of the hot spots for both botrytis and for sour rot. Make sure you're using a calibrated sprayer. Make sure you control powder and mildew, because I'm not convinced that powder and mildew is the culprit, but maybe if in BC you could be getting to that tail end of the fruit susceptibility period and getting those diffuse infections that are causing the wounds to the berries. Another thing you could try is cover crops. If you can have something that's taking up the water, if you have a deluge, then it would offset the amount of water that's going to be moving into the berries that's taken up by the light.
Any other questions? Oh, right up.
I forgot, what was the result on the looser clusters? Did that lower the incidence of sour rot?
I couldn't show a correlation between cluster looseness and reduced sour rot. But the GA and the high GA and the Prohex calcium I, they didn't necessarily all have looser clusters, but the sour rot was lower in the Pinot. So I'm not sure what's going on. Maybe it's my way of measuring cluster tightness that's off. Maybe it's the students that did the rating here in the back of the room.
Any other questions?
From the treatments, was there a reduction in Brix at all? From the gibberellins or the...
There was no significant difference among any of the treatments. Right.
Did the bloom time leaf removals, or did that actually affect the cluster thickness?
Not the way that it's been shown to happen in Italy. I'm really surprised, and I did talk to Dr. Tony when he was here at convention about that. And he said that that technique tends to work better with large clusters and very high cropping levels. So if the vine is carrying a lot, Pinot Noir, a little tiny cluster like this, Riesling, not much bigger. He's looking at Sangiovese and some of those big honkin' Barbera clusters that are going to be, that's what he was looking at.
Demanding more.
He actually had success with it in those. Brian had success with Vignoles, which is also pretty big cluster. I haven't given up yet. I have some other ideas yet.
Are there any other questions on that? Well, please join me in thanking Wendy for having a token of our appreciation for you. And just a reminder that yeah, a little mini. Just a reminder that our speaker next week is Dr. Ralph Brown from the University of Guelph, looking at new tools to fine-tune quality harvests. Look forward to seeing you there.